Hi, I'm Daniel. This video will explain DNA replication with a step-by-step process and a detailed diagram. This was made for Mrs. Bates' biology class. Before I explain the steps of DNA replication, there are things we need to know. DNA is made of nucleotides, which have three parts: a phosphate, sugar, and base group. The bases of DNA are adenine, thymine, guanine, and cytosine. The bases only pair in a specific manner: adenine with thymine, and guanine with cytosine. Without these base pairings, DNA would not be able to successfully replicate. Also, when DNA is divided, it is divided into two strands, a leading and a lagging strand. The lagging strand has fragments added to it, while the leading strand is being continuously synthesized. With that out of the way, let's get into the process. Step 1 of the DNA replication process is the DNA being unzipped or unwinded. The enzyme helicase breaks the hydrogen bonds, separating the bases of the DNA. Helicase only works because of topoisomerase, which prevents the DNA from being twisted and messed up. Topoisomerase is the enzyme that allows for a well-formed replication of DNA and helps helicase break through the weak hydrogen bonds of DNA. When the DNA is divided, both separated strands are almost ready to be made into two new DNA strands. There's still more that needs to happen. Multiple RNA primers are added to the lagging strand, along with pieces of DNA called Okazaki fragments. These are created by the enzyme primase and are added to the DNA strand through the enzyme polymerase. But there are two kinds of polymerase: polymerase 1 and polymerase 3. Polymerase 3 synthesizes the leading strand of DNA, and polymerase 1 synthesizes Okazaki fragments and RNA primers in the lagging strand. So DNA polymerase pretty much fixes up each DNA strand, allowing for them to be made properly. Meanwhile, during this process, binding proteins bind to each DNA strand, keeping them stable to prevent damage during the replication process. They do this by keeping the DNA unwound or unzipped during replication. DNA ligase is there to seal any breaks in the DNA strand, almost like it repairs the little mistakes that occur. Ligase only works on the lagging strand of DNA. After all of that is done, DNA replication is completed. You have two new strands of DNA ready for the process of mitosis. I would like to credit my partners in this project, Brant and Charlie, who helped with the art and research of the information. Thanks for watching.